This is the video lab report about the Infrared Spectroscopy lab by Colton Rathbun and Filippo Albertino. The purpose of this experiment was to determine which compounds, the formulas and the name of the compounds that were provided as unknowns. 2 liquids and 1 solid unknowns were provided. Some background information: the process of infrared spectroscopy is used to determine which functional groups are characteristic to each compound that's analyzed. This is done by determining through the infrared spectrometer at which wave number the functional groups are present. The wave number is symbolized with this letter, and that is the formula to obtain the wave number, 1 over wavelength. This can be done because of the characteristic wave number at which each functional group is found. The infrared spectrometer works by shining light on the bonds of a compound. The bonds subsequently get excited and release energy and start vibrating. The vibration is picked up by the device, and the frequency at which the vibrations take place is then analyzed and collected as wave number. This is the device that was used, the infrared spectrometer. The compound is placed on top of the sensor if it's a solid. In case of the liquids, it was placed on a salt plate and then inserted in the device. This is a list of the most commonly found functional groups. Some groups that were found very frequently were CSP3 and CSP2 and C double bond C at their characteristic wave numbers, which is around 3,000 for CSP3 and CSP2, and around 1,600 for C double bond C. The procedure: the first liquid was placed on a sodium chloride salt plate. It was just spotted with a couple drops. And then before running the procedure, a blank was actually placed in the spectrometer to calibrate the machine. Then the first liquid was analyzed by inserting it in the spectrometer, and the data was collected through the software that's connected to the device. The software then displays all the data that is picked up. The graph's y-axis were then changed to show percent transmittance, and the same procedure was performed for the second liquid unknown. For the solid, instead of placing it on a sodium chloride salt plate, it was just placed on top of the sensor as previously mentioned. This is the IR spectrum found for the first unknown, which was labeled as L2. Some characteristic peaks are found here. This is CSP3, CSP2, and C double bond C. Because of the presence of these three functional groups, the liquid was assumed to be 1 octene. As mentioned before, these are the functional groups that were found. 1 octene, which has a formula of C5H12O and a molar mass of 88.15 grams per mole. This is the structure. Here is the C double bond C, which also originates CSP2H on both sides of the bond, while everywhere else, every other carbon on the chain is CSP3. 8 carbons are found in the main chain and overall inside the molecule, and a total of 16 hydrogen atoms, which totals a molar mass of 112 grams per mole.
The second liquid that was known as the unknown was L7. The 2 major functional groups were found here for the CSP3H bond and here for the OH bond. The CsP3H bond was at 3,000 to 2,850 centimeters to the -1, and the OH bond was at 3,600 to 3,200 centimeters to the -1, which gave the unknown compound to be known as 2-pentanol. The formula was C5H12O with the molar mass being 88.15. Here are the CsP3H bonds that are in the chain, and then the OH bond is of course right here. With this compound there are 5 carbons and 12 hydrogens and 1 oxygen. So whenever plugged into the molar mass formula, you get the 88 grams per mole. The solid unknown was S6. The only functional group listed in the IR spectrum was here for the CsP3H bond. This CsP3H bond was at 3,000 to 2,850 centimeters to the -1, and it was assumed to be anthracene with the formula C14H10 and the molar mass of 178. As you can see here, though, anthracene has all these double bonds in all these various locations, which would only give CsP2H bonds. However, the molar mass is found to be 178 with the 14 carbons and 10 hydrogens. A major issue came into effect whenever trying to find what the solid compound could be. The most similar one was anthracene. The spectrum, however, as previously stated, only showed CsP3H bonds, but lacked CsP2H and C double bond C bonds, along with the benzene ring that would show anthracene. In order for it to be anthracene, you would need a benzene ring in this location, a CC double bond here, and a CsP3H bond here. However, that was not present. The opposing fact was that all the unknown compounds had CC double bonds and benzene rings. Therefore, our spectrum did not quantifiably show a specific unknown compound. However, all the other compounds beside anthracene had CO double bonds, NH bonds, or OH bonds, which were definitely not present in the spectrum either. So the most similar one was assumed to be anthracene. A possible reason for this, for the absence of these CsP2H and C double bond and benzene rings, percent transmittance and wave numbers were assumed to be because of a major shift of the spectrum. There was also consistently a small signal at 2350 centimeters to the -1 seen in this area right here. Right there, and it was possibly due to traces of previous compounds left on the device. However, the liquid signals gave very good IR spectrums and were able to be identified very easily. But it was determined that the IR spectrum might be better suited to study liquids than solids. In conclusion, though, the goal was met. The identification of unknown compounds using the IR spectrum was mostly successful, with the liquid unknowns being able to be determined, and the solid compound being able to be somewhat assumed based off of reasoning. This lab reiterated knowledge learned from lecture and applied it to the laboratory setting. It allowed for the new technology that the Science Center bought to be used in the chemistry laboratory, and it allowed for different readings and measurements to be done so that we could determine these functional groups and potentially determine the compounds that were unknown. Multiple literature sightings were made. Azonano was used to find the picture of the IR spectrometer. ChemSpider was used to draw the compound structures, and making the connections in organic chemistry was used for background information on IR spectrometry.